Now we are going to deal with that part of the gynoecium which bears the female gametophyte. Okay, so we are dealing with ovary over here. When we talk about ovary, inside ovary we have ovules. Inside these ovules you are going to find embryo sac, that is otherwise known as female gametophyte, and inside it you are going to find the egg. What you see over here is gynoecium. This is the stigma, this is style, this is the ovarian locule, it has been cut, and you are shown various number of ovules over here. Ovules are of different types, but what our syllabus asks us is to understand the structure of anatropous ovule. How an anatropous ovule looks like from the point of view where we can understand where is the location of gametophyte. Okay. Now, in the case of anatropous ovule, one thing you have to remember that there is a stalk on which the ovule is going to grow like this. Okay. There are going to be layers, and inside it you are going to find something that would be known as the nucellus, and inside it there would be an embryo sac, and inside it there would be the egg. Now, what happens is, supposedly you increase the length of this stalk, this ovule that was formed over here, it takes a 180-degree turn like this. So, what happens is, the opening that was present as I drew earlier, it is going to be present in this side, like this. So, the egg is going to be present over here, the gametophyte that I talked about. So, in a way, one condition is this, that the ovule is present over here. The other condition is I extend it and make it 180-degree turn. So, whatever was present at the tip at this side is going to be altogether opposite at 180 degrees, and that is what we have in anatropous ovule. Opposite to anatropous ovule is orthotropous ovule that I drew previously, and we have to study the structure of anatropous ovule that is given over here. See over here, this entire stalk has taken a U-turn of 180 degree. So, we are going to see in the ovarian locule, I would ask you to see over here, these ovules are attached to the ovarian wall by some agency, and that agency is known as placenta. What we have over here is, what joins the ovule to placenta is known as funiculus. Okay? The point at which the ovule joins with the ovary is known as placenta. How it is joining, what part joins the ovule with that particular site is known as funiculus. This funiculus joins with the integuments. Now, what are integuments? Integuments are the coverings. Okay? Integuments are the coverings of the ovule which would later on form the seed coat. This ovule is going to get converted into seed. Basically, we are seeing how ovary is going to form a fruit, and inside the fruit you find seeds. Those seeds have covering. You must have eaten pulses, they are seeds. You have seen that the pulses are having a covering over them. That is seed coat, and that has been formed because of these integuments. There lies an outer integument, there lies an inner integument. So, that part of funicle which joins with the outer integument of an anatropous ovule which has taken a U-turn is known as hilum. This funiculus has a central arrangement known as raft that extends up to chalaza. Now, what is chalaza? Chalaza is that point in the ovule which is exactly opposite to the opening pore known as micropyle. You see, ovule has to receive the male gametes which will come with the pollens. So, where from they are going to enter and reach the female gamete? This site, this particular pore where the integuments are absent, that is known as micropyle. Totally opposite to micropyle lies the chalaza. So, you simply have to remember that whatever lies completely opposite to chalaza is micropyle, or whatever lies opposite to micropyle is chalaza. Okay, raphe extends till chalaza. Then we have, after chalaza, this funiculus that had extended, it extends and takes a covering that is known as integument. Now, why these coverings are present? Because they are housing a nucellus. Nucellus is the arrangement where many types of diploid cells are present. Some cells of the nucellus, one of it becomes specialized, it undergoes meiosis and forms the embryo sac. This part, some cell of the nucellus. Later on, this nucellus would be consumed. Okay, before we come to the embryo sac, I want you to remember one thing. What is Errol? A-R-I-L. You eat lychee? lychee is Errol. Errol has stored fructose inside it, and that very tasty part is Errol. What is Errol? Errol is the third integument that often develops outside the embryo sac. Now inside this embryo sac you are going to find the cells, and one of them would be the female gamete. All of these would be haploid cells, rest of the embryo, rest of the ovule would be diploid. Housed in the nucellus lies the embryo sac, which is the female gametophyte that has the gamete known as egg cell. Now egg cell along with it has two synergids which enable the entry of male gamete to the egg. Apart from that, the central cell have secondary nucleus, and we have three antipodals. Now while remembering this diagram and while learning, you have to remember which side has micropyle and which side has chalaza. So the antipodals are always facing towards chalaza, while micropyle is always facing towards the synergids. This point is to be remembered. I would suggest you to practice this diagram more and more so that you get an understanding where from the seeds are being formed. Now next lessons that we have, we are going to see how seeds are formed, but where are they formed, that would be covered by understanding this diagram. Now I have tried my best to make you understand. You have to try your best by practicing it and learning what has been told.